Welcome everybody. Today we have Dr. Ranabhat talking about plant health problems affecting conifers. Dr. Ranabhat began his journey with the Entomology and Plant Pathology Division in July 2023 as an assistant professor at the Extension Plant Pathologist located at the Central Region Agriculture Extension and UT Soil Plant and Pest Center in Nashville, Tennessee. Jeff actually met him in Crossville. We do a lot of work in Crossville, Tennessee, which is on the way to Knoxville from Nashville. He's an extension plant pathologist and is dedicated to addressing key disease challenges in ornamentals, nursery plants, and turf. His research stems from direct input from county agents and growers, focusing on the challenges faced by growers. Okay, thanks a lot, Dr. Namar. It's all yours.
Good morning, everyone, and thank you, Jeff, for inviting me to speak about the plant health problems affecting conifers. I really appreciate everyone for registering and joining today's session. So I'm really excited to be here. All right, so as you already heard about in the introduction, I'm an assistant professor and extension plant pathologist in the Department of Entomology and Plant Pathology at University of Tennessee. Yeah, main campus is in Knoxville, so I am located here in Nashville, in the middle of the Tennessee. You know, Tennessee is very long, and maybe it is easier for me as a state extension pathologist to travel around the state. So as an extension plant pathologist, my responsibility is to develop a extension and applied research program on the disease affecting ornamentals, all ornamentals, landscape plants, nursery plants, and turf. You know, for more details, I have my lab website here. You can scan that QR code, so directly go to the lab website. That's our ornamental pathology lab. There you can find more details on what's my research and extension program, and also more importantly, you can find some publication there. And publication about how to manage and identify the disease of ornamentals. And there is a one section there like a plant health problems. When you click there, I put the pictures and short description where how to identify that, what causing that problems. I just started that few months ago, so it will build up in future, but there are some example you can find the disease, how to identify them. All right, and the main research that I am doing right now is managing rose rosite disease. You know that rose rosite disease is a really devastating disease in landscape roses and all cultivated roses. So this is a viral disease, and there is no cure. So we are trying to find out the resistance gene in different cultivars so that in future we will recommend some the cultivars that have a resistance gene so that the landscape will be safe from this virus. That's the ultimate goal. So, all right, if you need more information, go to that website. I'm not talking more rose rosite today, so let's go back to the plant health problems affecting conifers and evergreens. So while we talk about the plant health problems, why I using that phrase is because not all problems are diseases. So I need to here a little bit differentiate between the disease and other plant health problems. So there are two general big group of factors that causing plant health problems. So one factors are abiotic factors. These are non-living factors that causing damage or in plants, like environmental factors and the cultural practices that we use to manage the plants, like environmental factors like flooding, heat, freezing, and also like cultural practices like using herbicide that drifts to the another plant, and also too much and too little of anything we apply to the plants also can damage the plants. So these are all the factors, these non-living factors cause disease in plant. And another group are the biotic agents. These are the plant pathogen and parasite that cause plant health problems. So, while we're talking about these problems, so, that sometimes it is confusing between the damage or the caused by the abiotic non-living factors and the pathogen, the damage caused by the pathogen. So how we can narrow that down just as broadly? There are some sort of patterns we need to observe when you see these damage or problems in plant or in the field. So what to look for to categorize them as factors, the damage caused by non-living factors or the pathogen. So there is some patterns like if the problems exhibit a uniform pattern or damage over a large area or over the large part of a single plant, right? So uniform pattern means have similar damage, maybe one whole part of the plant, or you can see here in picture, you can see all of these trees has the similar damage. And that damage appear at a high percentage all at once. That means the plants or the field was totally healthy yesterday, then you next day you see the high percentage of damage, maybe more than 50% of the damage in a single tree or in the whole field, or what the some corner of many plants has that damage at once. And then when you observe these patterns, these problems has no further development over time. Mostly they stay the same, the damage you observe at the day one. So if these patterns, uniform pattern, high percentage of damage appear all at once and no further development, these type of damage mostly caused by abiotic factors. So these are the abiotic disorders. So example, you can see the frost damage. Now the temperature is really low. You can see some sort of frost damage. So it observe the uniform pattern, like the tip of these spruce is browning here. It's a frost damage. Then you can also see in the summer like a drought stress. So this part of the plant is had browning. So these are the drought state. And also very common in many conifers is the winter desiccation. When the temperature drop and has a large, the windy condition, and also that summer desiccation also can cause in a hot and dry summer with windy conditions. In that case, what happened? In the winter, maybe the soil temperature drop down and the water froze there. Then in windy condition, then plants transpire faster than that of the root pull up the water. Then so that way that new growth of the tips of the conifers dried out. So in the winter windy condition as well as in the summer windy condition. So in that condition, you can observe these damage scars in the new growth. These are born out, and we can see these damage. So what we can do for the management wise overall for the damage caused by abiotic factors, these weather patterns and other desiccation. So what I can do, because these are the damage you already observed that at that condition, the immediate action is, what we can do, we can just wait and see there, like there is no any cure you can apply immediately to cure that damage, right? So in that condition, maybe, like sometimes you can supplement watering if the condition are dry. If it's still low temperature, that watering may not help, it will froze down there. And then pruning, right? The removing the dead parts is always important, because these dead parts, um, in the dead parts, maybe the other fungus and also the insect can come and infect, and that way it causes further damage to the other remaining part of the tree. So that's why I need to prune that disease part, but need to wait until the new growth appears, So to see how, to guide your pruning, you can do a scratch test where you can see the green part, green tissue indicate the living tissue and stem, and then the brown part is the dead tissue, right? Then we can prune the dead part, and then we can see the new growth from there. So management-wise, all the some cultural and silvicultural practices we can follow to deal with this type of damage. All right. Also, we can see some herbicide damage, right? Sometimes when we apply to one part and then that drift to the another part. So here is some example here. You can see in the picture the herbicide Imperial's injury. You can see some funky growth there due to the damage or injury from this herbicide. So in that condition, the plant may revive or resume their normal growth, but it takes time. So maybe a few years, the big tree can resume, but it takes time. So management wise is there is no any cure of that we can do. All right, so we talk about briefly about the problems caused by abiotic factors or non-living factors. Now we can switch gear to the problems caused by biotic agents, or normally we call disease. So like the patterns, we talk about the damage caused by abiotic factors. There is some sort of pattern also in the damage caused by biotic or agents or pathogens. So these damage caused by biotic agents or pathogen exhibit a random pattern in an individual plant or in the field. So that means maybe one plant in the field from one corner infected, then the other one in the randomly. Also maybe some branches or some needles in a tree infected severely, or like there is a random symptoms you can observe in a single tree. Maybe some part here in some branches, maybe in the other part in some branches, like a random pattern, not the uniform. That's the main things to remember. And the problems appear at a low percentage at the beginning, because that maybe a single spore or few spore infect that tree, right? So that starts from, small or low percentage, and then it progress over time. So you can see here in the picture from July to August, right? In July, you can see some infection here in leyland cypress. It's a Ceridium canker here, some damage, but after a few months, you can see how this disease progress, right? So this disease progress is due to the fungus grow more spores. Then these spores infect the other healthy part of the plant. So that's why these problems are progressive over time. So we need to observe our plants regularly to observe this type of pattern to record. Then we can broadly identify, oh, this may be caused by pathogen or maybe some other non-living factors. So you can see also here the disease progression from January to June. You can see how much damage we can observe due to the pathogen infection or disease. Okay, there are few pathogen groups that infect conifers, mostly the fungal pathogens, and then some oomycetes we also call water molds, and some nematode also infect conifers. And I think you also observe some parasitic plants, the mistletoe also infect these conifers. So these are the pathogen groups. There are a lot of disease, several species of fungus infect the plants. So I'm not going to talk about all of these, but I have some common pathogen that I will share today. So major disease, needle cast and blights is sometimes it is sometimes it is a needle blight or needle tips blight or needle cast. Is the common generic name caused by several pathogens. Then another disease or canker disease are really common, and root rots and the rust are the easily identified pathogens. So let's talk about the needle cast. These needle cast caused by a diverse group of fungi. There are several fungi can cause disease in several conifers. The main symptoms are of needle cast is characterized by the premature needle shedding. So not in the regular natural shedding, right? So prematurely in different season, not in the fall season or where the time the natural fall shedding come. And then these pathogens, these fungal pathogens that causing needle cast start from bottom of canopy and then progress upward. So mostly these fungal pathogens may overwinter, or they are in the base of the trees, then they slowly infect from bottom to the top. So these are some of the common pathogens that cause disease in several pine, fir, spruce, and these are the several pathogen name here, like rhabdocyline needle cast, rhizopharia needle cast. These are the common one we observe when we culture. In the samples come, or we collect the sample, we culture in the lab. So whatever the fungal pathogen causing different needle cast in different varieties of conifers, the general management practice, cultural practice are the same. So always we need to start from the clean, right? Use the disease-free seedlings is a common. We need to follow that, whether you are planting for few trees in your backyard, or you can use them as a commercial production, right? So disease-free seedling, then always, like these fungal pathogen need a high relative humidity, and so to if the plants are close enough, then the relative humidity increase. That's why the low air movement between the trees. That's why maintaining the proper spacing between the trees and controlling weeds can reduce the disease infection. As we all know about the disease triangle, right? There's three factors we need to cause the disease. One is a pathogen that can cause the disease, another definitely plant host, and then the conducive environment. If the environment is favorable, then the pathogen can infect the plant. So then, removing the severely infected trees and or branches that reduce the spread of the spores, then the other trees or the other part of the plants stay safe. Always, if we are always recommending to use the resistance cultivar if there is available, and also sometimes we need to find alternatives, right? Sometimes some conifers are severely susceptible to the one part of pathogen, like a Swiss needle cast in Douglas fir is really high risk, but if we can switch that to the Western hemlock or red cedar, maybe that way can reduce the infection. And I always recommend for the protective fungicides shortly after the new growth. But for the fungicide application, always need to read the labels. We need to follow the labels by to use the right dose, right frequency, and I also recommend to rotate between the groups of fungicide so that will reduce the resistant development on any type of pathogens. All right, so let's change a little bit on needle cast to the needle blight here. So these are also the several fungal pathogens causing blight, tip blight, mostly in several Juniperous species, Pinus species, and these are also these common as like a needle cast. For example, I'm going to talk about juniper tip blights here. The infection, there are two types of tip blight. One is causing by Pomixi blight. The another pathogen also cause the Cabatina juniperi, also cause the disease in juniper. These are the two different fungal pathogens that appears in different season. So the Pomixi blights appear in the infection occurs in the spring and late summer, and mostly started off from the lower branches. These when the pathogen infect, the typical symptoms is the scale turns yellow and die, and then the foliage become gray and develop a small spot. You can see here in the picture also. These type of symptoms are really common and causing by Pomixi blights and Cabatina blight. And at the point of infection, you can see some sort of dot and different fruiting bodies of these pathogen produced there, like Pomixi blights produce pycnidia and Cabatina blight produce acerbuli. This may be not that much important as a homeowner for me, but for the identification purpose, these are the main characteristic. So the Cabatina blight, the symptoms appear in early spring, maybe March to May is the season's coming up, then the Cabatina blight, but the Pomixi blights comes into the fall. So there is one publication here I published this year, Pomixi blight of juniper. You can find, this is a QR code for this publication. Also, you can go and search in the UT extension publication. The number is W1311. You can find the whole articles there, the details. For the cultural controls, like whatever tip blights, any fungal pathogen causing, there's a normal general management practices are the same, like using a resistance cultivar, inspecting the damaged branches, always start clean, and pruning the dead and infected branches. Then morning irrigation is important because most of these fungal pathogens for any ornamentals need more than 8 hours to 12 hours of leaf wetness to cause infection. So that's why when we reduce that duration of leaf wetness, then we can reduce the infection. When we irrigate in the morning, then the sun come up and then dry out the leaves, right? So therefore morning irrigation is always recommended practice for the irrigation. So after cultural practices, there are some chemical fungicide are also labeled for the fungal pathogen, these fungal blights. Here I also group them as a preventive or curative, and also for the commercial purpose and for the homeowner. So you can also have this information on that publication. And this FRAC code here, because these are the different group of the fungicide. So we always recommend to alternate or rotate with different group of fungicide to reduce the resistant development in the fungal pathogen. So this type of general cultural and chemical management practices can be used for different blights and tip blights and needle blights. That's another fungal pathogen is the Dothystroma needle blight. The main characteristic symptoms to identify this needle blight is banding here. You can see half of the needle is crossed because half remain green, then in other half you can see this band. Definitely we need to identify in the lab, but the general symptoms wise you can identify that way. And heavy shading in the inner branches from lower. When the plant is severely infected, you can see whole trees is the symptoms develop from bottom to up. All right, we talk about the needle blight and cast. Let's move to the canker disease. These are also very common in conifers. These canker disease are the localized infection. Sometimes is developed into the sunken necrotic lesion. Sometimes there is a point of infection only. You can observe at the beginning of infection. Then later, some pathogen develop different structure like the sunken area and necrotic lesion you observe mostly in branches and trunk. And when that pathogen infect from that branch, then is girdle the branches, and then they block the food and water movement so that the branch dieback occur. And mostly these canker pathogens are following the stress. When plants are weak, then they infect and requires a wound to establish. So that's why we need to be very careful on handling plants during planting and other part, because if there is a wound, then the fungal spores are everywhere, right? So they land there in the wound and then enter through that and infect the plants. There are several canker disease caused by different fungal pathogen. So, these are the some common example like Botrytisperia canker, Schiedium canker, Neonectria, Thionectria. These are the canker, the really common canker that infect these conifers. Well, for the canker disease, general management is the pathogen need a wound to infect, so minimizing wounding, handling or any time of the plant that may reduce the infection. And yeah, the pathogen follow the stress plants, right? Therefore, maintaining plant vigor with the regular irrigation and fertilization might help. If the plant is really strong and healthy and no wound, then the pathogen dare to infect. If we see a single branch or, at the initial point, then pruning that or destroying that infected branch save other part of the plant and other trees. Yeah, sanitation is always important, right? When we use some pruners or shears for pruning the branches, infected branches, then disinfecting them, then we can reduce the spread of the pathogen from one branch or one plant to another plant. For this canker disease, copper-based products may help, but normally when we notice the severity of the disease, is chemicals are not that much helpful at that condition, cost-wise and management-wise. You can see this picture at the beginning also. This is Ceridium canker and Leyland cypress. So the main symptoms of this Ceridium canker is the lateral branch dieback. You can see these branches dieback from the point of infection. And then the another characteristic of this canker disease is the resin flow from the point of infection. You can see in this picture from this is the point of infection. And then from there, you can see these resin flow. That's the characteristic symptoms. Yeah. Look for the resin flow for diagnosis. And when we got a sample also, we look for that. All right. So these are the canker disease, uncommon. Let's move to the another pathogen type, this rust disease. So rust are really common and easy to diagnose. And most of these rust pathogens have an interesting life cycle. Most of them need more than one host to complete their life cycle, and characterized by yellow leafy spot and rust-colored pustule in different plant. They are, as I mentioned, most of them need two hosts or three, mainly two hosts, like from conifers to broadleafs. So these two hosts, different type of hosts, need to complete their life cycle, and they cause gall mostly in the conifers and in the dieback in the other host. So most common is cedar apple rust. Is very common. We can see that infect juniper and the apple, true apple, or the crab apples. These are large annual galls develop in the conifers, in the junipers. So another type of rust is the cedar hawthorn rust. These are tiny annual gall, like a very small tiny galls you can see here in the pictures. The cedar apple rust is really big. You can see like a brain shaped galls, but these cedar hawthorn rust is a very tiny annual galls they develop in the junipers. And cedar quince rust is another can cause canker. This is a perennial canker. Is a infect the plant stem. Is not like a gall forming, but is a laterally in the spring. But you observe these pustules, orange colored pustule in the stems. They are perennial. They remain for more than years. They this is a common one. You can see this life cycle here. You can see observe these galls in junipers in the fall. When the rain comes, then these galls develop tentacles, like yellow colored tentacles, and these tentacles produce millions of spore. And if you have kids, sometimes when bring that galls and put into the water at home, and they slowly develop these tentacles, and kids really love that. And then these spores, these called teliospore, they produce the teliospore, and they germinate into the different type of another spore called basidiospores. And these basidiospore infect the crab apples or apple. Then now in the April, May, we see lots of crab apples infected with these spores. So this is how then from these crab apples in the fall, this spore spores infect the juniper. That's how the life cycle complete. If nearby there is no another alternative host, maybe the life cycle just stay there. Management wise, yes, always recommend for the host resistance. There are some cultivars, they are resistant to these pathogen like crab apples. You can see some of them are here, and also some of the junipers are resistant to these rust. Cultural controls, sometimes the bloom thinning in apple production might help. Removing galls, yeah, but if there are, sometimes you can find hundreds of galls in a single tree, but it is, is it really worth to remove them? Depends, right? If you have many junipers and many galls, maybe it is not. I mean, time-wise, it's difficult to remove all of them. But if you see a few, is removing them definitely a good idea to control that. So some of the steroid inhibitor fungicide are used to apply for the other host, like apples and crab apples. These fungicide help to reduce the disease. Okay, this is the strong gall in you can see in pine. These galls appears in May and July. This you can see these orange color spores produced in these. And for this strong gall rust, the another alternative host are the oak trees. Then the life cycle complete like that. Another gall like a western gall rust that complete the life cycle in a pine tree only one host. But most of these rust need the secondary host to complete the life cycle. But the primary host are the conifers. All right, we talk about blights, tips, cankers, and then now we are switching gear to the root rots. Most common root rot are the Phytophthora root rot. These are the water molds causing really devastating disease on all conifers. So infected plants, as like wilted and stunted due to the infection in the root. And when you can see in the picture also the discolored root here, decaying root, right? So that the not normal functioning disrupted by that, and then the plant turned wilt or stunted. So these are the water molds. They love water. So standing water and poor drainage is a really conducive environment for them. These, if we can make unfavorable condition, then we can reduce the disease, right? So you can see these, sometimes you can see the whole plant dying, maybe start from the bottom. Sometimes you have a stem canker also. This Phytophthora is a really devastating disease. You can see whole field from the production site also infected with Phytophthora. So how that pathogen spread from one field to another field, maybe the infected seedlings, that's why you need to start cleaning, right? And contaminated soil also. Sometimes if you have a production site, then from the equipments, then we can transfer from one field to another field, and also the infected water, right? From one field to the another field. That's how the pathogen loves water. They have a spore, they swim in the water and move slowly. So you can see how the root damage here, root rot causing by this Phytophthora pathogen in pine tree. In fact, almost all of these type of conifers really devastating disease. You can see this example here. In the leyland cypress, you can see this root here. Yeah. So whatever the part of the plant infected, like root, shoot, or stem, how we can manage these water moles, phytophthora. So mainly cultural practices are really huge. Starting the healthy seedlings, choose resistance or tolerance varieties if they are available. Always look for that option from the beginning, and avoid planting in a poorly drained soil. That's why for a homeowner also, if they are looking, planting that, site selection is really important. And improving soil drainage also help to reducing that infection. And as it spread from one field to another field by soil transferring infected soil to the another field, so preventing that also reduce the disease infection from one field to another field. And some of the chemicals are also available to manage this disease. All right, so this is the sample we got in the diagnostic lab, and our diagnostician consulted with me. After that, we identify root rot here. This is the another root rot. Is the Enosus root rot in Cannon field. You can see this Christmas tree plantation. Some of them are browning already there. Then the sample came into the lab, and you can see the root rot here. So these are another example. You can see some of these trees. These are the stump from the previous Christmas tree, then nearby tree is infected. Anosis root rot is caused also by a fungal pathogen called Heterobasidium anosum. And this fungal enters through the wound and then goes to the heartwort and cause decay the root system. Then wilting appear later, right? So maybe then there is an interconnection between the roots so that the nearby tree also get infected. So mostly the mortality came into the second and third year. It's really destructive disease. I like to shear here, then we identify that and then we consult with the grower, hey, we find out that pathogen, then the growers came like, oh, okay, then what next? So the growers ask me some question and then I consult with my answer. Maybe I feel like that may be useful here to present so that how that is the process you also aware of, and then that might help to deal with the pathogen. So the grower asked me, should we apply a fungicide treatment in the field after identify that pathogen? Yeah, maybe if it is in person, I'll ask you that question here in Zoom. Okay, that's my answer. Yes, fungicide treatment in the field may not be cost effective at this condition, as a lot of the trees are already infected in the soil level. Okay, another question is, should we apply a preventive fungicide treatment on the tree that are not affected in the field? Think about that. Yeah, possibly, but if the pathogen, we don't know how much that pathogen already spread through the root connection in the soil, then may not be cost effective. Yeah, right. Maybe the symptoms may not appear in the above ground part. So it is hard to say that, but yeah, maybe possibly we can use that, but always need to calculate the cost of that. Another question, can we replant any new tree seedling next spring in the same areas? That's not a good recommendation to do that. There is no data to determine how long the fungus remain in the soil. It is hard to recommend that. And also another question, should we plant the same variety of field tree in that area? Planting the same variety of field is not advisable because that may infect the soil. The fungus may remain in the soil. So for the stump infection, the borax can use because the fungal spore infect that cutting wound of these stumps. So that's why removing all these parts, cut stumps, and it's a good idea because you can see in the field here, remove all the trees and stumps remaining part infected with the, after selling, harvesting the Christmas tree. For the management wise, resistance plant is always a good option, and always monitoring if there is any possible infection there, and treat the freshly caught stumps to prevent the spreading infection. All right. So for the success in disease management, I always use this analogy of this iceberg. You can see how big iceberg in the top and how bigger is in the below the water. So when we apply our strategy before the problem occur, then we have a greater success than that of the strategy we use after we notice the problems, because after we notice the problem, there is limited option available for curing that pathogen. Sometimes it is too late for the effective treatment, and sometimes it is not available, any treatment, right? And also, if we use a lot of chemicals, then development of the resistance on that pathogen and may not cure later. So that's the secret. So we need to start from the beginning, from the site selection, variety cultivar selection, and regular monitoring, right? We need a integrated plant disease management strategy to manage any type of disease. So there are some components, for example, selection of the resistant varieties of, if available, and go for that. And then after that, we need a cultural practices, right? Regular watering or fertilizing and whatever, make that plant bigger, sound. And then monitoring and scouting. If you plant and left, maybe if you don't care, that's not a good idea, right? So that's why you need a regular monitoring, a regular care, scouting. If there is any change come up, that way that helps a lot to early and accurate diagnosis is, right? If we early identify that disease, maybe one branch is infected first, always, right? Then we can save that tree, other tree. So that's why early, and also we need accurate diagnosis to manage any disease. And then the last option is chemicals. Always read labels if you are using any chemicals on that. All right, that's what I have for today, and thank you so much for you joining today. And here is my information. If you need, have questions, it's my email address, and you can follow me in X also. And I'm posting what's going on about the disease-wise in ornamentals. And here, if you have any question, I'm happy to answer. Thank you.
Nar, can you take a question or two from the group?
Sure.
Okay, I think there'll be a couple of other questions, but I had one which was about the borax.
Okay.
Can you elaborate if you use that only on stumps to stop spread in a field that you've cleared, or how would you apply it to trees, not stumps?
Okay, that's a great question. So that fungal pathogen attract by the freshly caught stump, so that then if that is in the air, that the fungal spore fall into the stump and grow there. That's why the recommendation is to apply borax on the stumps, not in the unwounded tree, not in the healthy tree. So if there is a wound, yes, you can apply there, but the main recommendation here, the borax is for the stump.
Okay.
Yeah, cutting part, so that the fungal spore won't grow there.
Another question came in about, are there specific diseases that you're aware of that occur to the genus Araucaria?
So For now, in top of my head, I'm not sure that, do you know that genus common name of that? I mean, I'm asking.
It doesn't grow here in this part of the
Okay.
country. So
Okay,
yeah,
so I'm not aware of that. Maybe if you need, please email me. I'll have a look on that, you know. If,
Somebody did have a species other name last
yeah,
week that I believe.
it's the monkey puzzle tree that
Got
yeah
it.
that most people can't grow. It's very susceptible to root rot, I think.
okay. Yeah, thank you for that.
Um,
Oh, and Norfolk Island pine too.
Okay.
oh, somebody says, oh, the guy that's asking is from Australia.
Oh, okay.
That's why we're struggling a little. I mean, I've seen those plants, but they don't grow real well here.
I see. Okay, I'm stopping here, then we
I
can.
think Warwick is from Australia. I think he has a nursery
Uh,
in Australia, if I remember right.
he tried to ask those questions last week too, and we're like,
Another question about Japanese maples and powdery mildew.
Oh, okay. So, in the lots of information, okay. Um, what was the specific question for the power limit? Oh, how and when does one treat establish Japanese maple for? So, powdery mildew is the only one fungus that do not require water or the relative humidity to infect. So year around there is a one publication also for me on the UT extension. You can go for more information on that. So normally protective fungicide we can use after the new blooms, and then depend upon the how severe the disease is. Normally I do not recommend to apply unless you have a severely infected plant. So these fungal pathogen do not kill the plant. Yeah, depends how big is the plant is. So for the Japanese maple, if you really have a good, I mean, you really love and have a very frequent infection, you can use some protective fungicide for the powdery mildew.
I was excited to see that you showed pictures of apple cedar rust gall. Um, I didn't know what that was until we moved to Tennessee, and they're quite prevalent here, and they can get quite large, almost the size of baseballs. And then they have that oozing stuff that comes out. It's quite an actually exciting sight to see until you realize what it is.
Yeah, totally. Yeah, really weird structure there. You can see from the tiny galls to the bigger one. Yeah, and then that rains, the water stimulate to comes out these tentacles.
Yep.
Yeah, and produce this spore and infect the apple species.
Yes. But we didn't know that, so we planted apple trees, not realizing that our property was surrounded by um, native cedars that carry
Yeah.
that gall. And yeah, so we don't really have productive apple trees.
Okay. Yeah, these, and if they are close by, definitely they are there to complete their life cycle, right?
Yep. So we gave them the life cycle.
Yeah, yeah, we give the chance to come. You give the chance to complete that. But like they are everywhere, right? I mean, there are thousands of eastern and junipers around, and apple, and they produce millions of spore.
Yep.
So, and travel very long distance, kilometers, miles of miles. So, is not. I'm not just blaming you. You can get from anywhere.
So people can unmute their mics if they want, or they can put their questions in chat. Um, Do we have any more questions? Well, while we're waiting, if in case anybody has any more questions, make sure everybody goes to our website and signs up for the national meeting. It's in June this year in the Quad City area. So that's like Illinois, don't ask me, out there. It's out west somewhere. It's a great meeting. They have unbelievable gardens there. So make sure you're going to the website and looking at that. It doesn't look like we have any more questions. I want to take a chance to thank you. It was very exciting to see all those diseases. We all are inflicted by those, especially root rot is a problem for us. And some of I mean, I've seen it all, but it was very enlightening. So thank you again. Anybody else? Deborah, do we have any more questions?
No, I don't see any more.
Okay.
I think, you know, it's heavy last week with pests and this week with pathogens. Sitting here when most of us are dormant. So I guess we just have to be Sherlock Coneheads in the spring. Yeah.
Yes.
Or with the copper fungicides, is it best to spray those like in the spring and fall, or can they be sprayed like early morning in the summertime too?
I think these are mostly the protective one, and mostly it's better to spray when the new birds come, so that time, and then depend upon that, whatever you buy, then we need to read the labels, right? But normally recommend from the first flush out, and then when you see a little bit bird, and then from there, then we can go. Normally,
Yes,
protective one.
it's amazing how much information you can get from reading the label, right?
Exactly. Like what? Yeah, totally. Otherwise, it is hard to tailor with the particular fungicide, right?
Yes, for sure.
Yeah.
Awesome. That was great. Thank you. We appreciate it. And we'll be talking to you about some other things that we can do together with the Conifer Society and your department in the near future.
Totally. I really appreciate, and to be here, delighted to be here and talk about the disease. And I am open to work with the society. Yesterday we also talk about that, and I'm happy to work on some way or other way, to do some research on conifers, collaborating with the society. And thank you for inviting me.
That's great. Thank you. Very good lecture. Appreciate it.
Thank you, Norm.
Thank you.
You're welcome. Thank you. Bye.
Bye, everybody.